The topic of the poster I am presenting is the synthesis and molecular modeling of purine ribonucleotides as potential ligands of the human G-protein coupled receptor 17. My name is Marco Rabuffetti. I am a postdoc fellow at the Department of Chemistry of the University of Milan under the supervision of Professor Giovanna Speranza, and this work is being carried out in a collaboration with our research partners at the Department of Drug Sciences of the University of Pavia and the Department of Biomolecular Sciences of the University of Milan. The research I'm going to present focuses on the human GPR17 receptor, which is a G-protein coupled receptor usually overexpressed in damaged brain tissues. As a result, it is a potential target for the treatment of diseases caused by neurodegeneration, such as multiple sclerosis. Both agonists and antagonists with different structures are well known, and some examples are represented here. It is very important to know that many of these molecules have a nucleotide structure. Now, since the 3D structure of GPR17 is not known to date, we used a homology model based on another receptor, P2Y1, which is phylogenetically related to GPR17, in order to perform docking studies on a library of purine nucleosides and nucleotides. From the docking score profile, we identified the 4 best potential ligands, which are represented here, that share 3 common structural features: a 5' phosphate group, a substitute purine base, and an isopropylidine group. Then we proceeded to synthesize these 4 compounds. Specifically, the first product, 8-methylaminoinosinic acid, was obtained by bromination in position 8 of inosine, followed by halogen substitution with methylamine. Concerning compound 2, the strategy we followed was to activate the exocyclic amine in position 2 of the purine ring to form a bromoderivative and displace it with N-octylamine. On the other hand, the N2 acylations necessary to synthesize products 3 and 4 were performed using the proper acyl chloride or anhydride through a transient protection strategy. All intermediates were subjected to direct phosphorylation to form the final products. Finally, the 4 synthesized nucleotides are going to be tested soon in binding assays by surface plasmon resonance, SPR, a technique which enables the measurement of binding affinities and kinetics. Briefly, GPCRs are typically very unstable out of the cell membranes, so 2 engineered variants of GPR17 were extracted from crude membranes and immobilized on the surface of an SPR sensor chip. The validation with the antagonist Cangrelor and the agonist Azinex1 allowed us to demonstrate that SPR is a very reliable tool for the identification of agonists and antagonists of GPR17. I would like to thank all the research groups who have been involved in this project, and if you would like to learn more, feel free to contact me during the meeting. Thank you.